बिस्मिल्लाहिर्रहमानिर्रहीम। अस्सलामु अलैकुम रहमतुल्लाहि व बरकातुहू। रब्बि ज़िदनी इल्मा। अल्लाहुम्मा इन्ना नस्अलुका इल्मन नाफ़िअ व अमलन मुतक़ब्बिला व रिज़्क़न तय्यबा. We are discussing the biochemistry of proteins and we are on the topic of classification of proteins. Several classes are discussed. Today we are discussing the derived proteins. Derived proteins are those proteins which are derived from either simple or conjugated proteins, and these proteins are produced by various physical and chemical factors. They are divided, so mainly two factors are there on the basis of which they are further classified. We have primary derived proteins. These are denatured or coagulated proteins that are placed in these group. Their molecular weight is same as the parent protein, but they differ in their solubility, in precipitation, crystallization, heat, X-ray, UV radiation, vigorous shaking, and other physical properties are different from their primary proteins. These are the primary derived proteins. There is intramolecular rearrangement of the primary protein that lead to the changing in their physical characteristic. Primary derived proteins are synonymous with the denatured protein in which the peptide bond remain intact. Peptide bond सही रहता है, लेकिन बाकी मॉलिक्यूलर की अरेंजमेंट हो जाती है, रीअरेंजमेंट हो जाती है, इसलिए स्ट्रक्चर इसका डिफरेंट होता है प्रोटीन से. एग्जाम्पल्स हमारे पास प्रोटीन्स. These are the insoluble products formed by the action of either water, very dilute acids and enzymes, and they are predominantly formed from certain globulins. For example, myosin from myosin, elastin from elastin, and fibrin from fibrinogen. Fibrin is a popular protein जो कि basically derived protein है. Then we have metaproteins. They are formed from further actions of the acid and alkalis, and they are generally soluble in dilute acid and alkalis. They are insoluble in neutral solvents like acid and alkali. Third type is the coagulated proteins. They are coagulated proteins that are formed by insoluble products by the action of either heat or alcohol on native proteins. Examples are, for example, cooked meat, cooked egg, albumin, and the example includes cooked meat, cooked egg, albumin, and alcohol that precipitate the proteins. Then we have secondary derived proteins, and these are proteins formed by the progressive hydrolysis of the proteins at their peptide linkages. They represent great complexity with respect to their size and the amino acid composition. They are roughly called as proteoses, peptones, and peptides according to their relative average molecular size. It means कि अगर हम differentiate करें primary derived proteins को secondary derived proteins को, तो secondary derived proteins के अंदर जो peptide linkages हैं वो भी hydrolyze हो जाती हैं. Proteoses and albuminoids, these are the hydrolytic products of the proteins which are soluble in water and they are coagulated by heat, and they are precipitated from the saturated solution of the ammonium sulfate. Then we have peptones. These are the hydrolytic products of the proteoses. Further इसमें hydrolysis हो चुकी है, further peptide bonds टूट चुके हैं. They are soluble in water, not coagulated by heat, not precipitated by the ammonium sulfate. They can be precipitated by, however, phosphotungstic acid like products obtained from the enzymatic digestion of the proteins. Peptides are composed of only a small number of amino acids joined as a peptide bond. अब क्या है, protein chain short हो गई है. They are named according to the number of amino acids present in them. Dipeptide may be there, tripeptide may be there. They are water soluble and usually not coagulated by the heat and are not salted out from the solution. However, they can be precipitated by phosphotungstic acid. The complete hydrolytic decomposition of a protein generally follows the stages given below. For example, protein is hydrolyzed into metaproteins and then proteoses, amino acids, then peptides, and then peptone. And peptide and peptone are the interconvertible. The products from protein to peptone give a positive Biuret reaction and are relatively large molecule, while dipeptide and amino acids do not give Biuret test positive, and therefore they are also called as Biuret products. Now the summary of the derived proteins. These are the either enzymes, they are body catalysts, they are contractile muscles like myosin, they may be hormones, there may be neurotransmitters, eupepellins and endorphins, they can be storage proteins, they can be transport proteins. Primary derived proteins are the denatured proteins, while secondary derived proteins are polypeptides, oligopeptides, proteoses and peptones. This is all about the classification on the basis of the hydrolysis of the protein. Thank you.